Hey science fans! Welcome to another exciting episode where we unravel the mysteries of science. Today we're diving into the fascinating world of karyotyping. What's karyotyping, you ask? Well, it's a bit like peeking into the blueprint of life itself. Imagine it as reading the instruction manual for your body. Just like a manual tells you how to assemble a piece of furniture, karyotyping tells us how our bodies are put together. You see, our bodies are made up of trillions of cells, and each cell has a nucleus. This nucleus is like the control center of the cell. Inside the nucleus are chromosomes. These chromosomes are crucial because they carry all the genetic information that makes you, well, you. Think of chromosomes like tiny scrolls that hold your genetic information. They are tightly coiled packages of DNA, which is the code that determines so much about us. They determine everything from your eye color to your height. Pretty cool, right? But that's not all. They also influence many other traits and characteristics. Karyotyping allows scientists to examine these chromosomes up close. By doing so, they can see the detailed structure and organization of our genetic material. It helps them understand how they're structured and identify any potential problems. For instance, they can spot abnormalities that might lead to genetic disorders. Now, you might be wondering why we need to look at chromosomes so closely. Well, the answer lies in the importance of genetic health. Sometimes there can be small changes or rearrangements in these chromosomes. These changes can be as minor as a single gene mutation or as significant as an entire chromosome being duplicated or missing. Even tiny changes can sometimes cause health problems or genetic conditions. These conditions can range from mild to severe and can affect various aspects of a person's health and development. By studying chromosomes through karyotyping, scientists can diagnose and understand these conditions better. This knowledge is crucial for developing treatments and interventions. So let's explore the amazing journey of karyotyping together. Join us as we delve deeper into the science of chromosomes and uncover the secrets they hold about our health and identity. So, how do scientists get their hands on these tiny chromosomes? It all starts with collecting a small sample of cells. This could be a blood sample, a piece of skin tissue, or even amniotic fluid for prenatal testing. Don't worry, these procedures are usually quick and relatively painless. Once the sample is collected, it's whisked away to a lab for the next exciting steps. Imagine those cells going on a little adventure. Just like we need food and water to grow, cells need a special environment to thrive. In the lab, the collected cells are placed in a petri dish containing a nutrient-rich solution called a culture medium. This medium is carefully prepared to include all the necessary vitamins, minerals, and other nutrients that cells need to grow. Think of it like a delicious soup that provides all the essential ingredients for cells to grow and multiply. The culture medium is like a gourmet meal for the cells, ensuring they have everything they need to stay healthy and active. Once the cells are placed in this nutrient-rich environment, they are kept warm and cozy in an incubator. This incubator acts like a tiny oven for cells, maintaining the perfect temperature and humidity levels to mimic their natural habitat. The incubator ensures that the cells are in an optimal environment, allowing them to grow and multiply efficiently. Over the course of a few days, the cells begin to divide and multiply, creating a larger population. After a few days, the cells multiply, creating enough material for scientists to study their chromosomes. This process is crucial for research as it allows scientists to observe the cell's behavior, understand their genetic makeup, and conduct various experiments to advance our knowledge in fields like genetics, medicine, and biology. Now, it's time to get those chromosomes out of hiding. To do this, scientists use a clever trick. They place the cells in a hypotonic solution. What's that, you ask? It's a solution with a lower concentration of solutes compared to the inside of the cell. Remember that water likes to move from an area of high concentration to low concentration. So water rushes into the cell, causing it to swell like a balloon. Eventually, the cell membrane can't take it anymore and bursts open, releasing the chromosomes. Chromosomes are super tiny, even under a microscope. To see them clearly, scientists use special dyes like Gemsa or Wright stain. These dyes are like magic markers for chromosomes, staining them different colors based on their structure. The dyes create a beautiful banding pattern on the chromosomes, making it easier to identify each one. It's like giving each chromosome its own unique fingerprint. Section 6. Spreading out the instructions, preparing the slide. Now that the chromosomes are stained and visible, it's time to put them on a slide for viewing under a microscope. Scientists carefully spread the chromosomes onto a glass slide, making sure they don't overlap too much. This step is like organizing puzzle pieces before you start putting them together. It ensures that each chromosome can be seen individually. Section 7. Looking for clues. Examining chromosomes under a microscope. The moment of truth has arrived. Scientists use a powerful microscope to examine the stained chromosomes on the slide. They look for any changes in the number, size, or shape of the chromosomes. This is where the real detective work begins. By carefully observing the chromosomes, scientists can identify any abnormalities that might be present. Section 8. Organizing the picture. Creating a karyogram. Once the chromosomes are examined, scientists arrange them in a specific order to create a karyogram. A karyogram is like a family portrait of all the chromosomes in a cell. The chromosomes are arranged by size from largest to smallest and paired with their matching partner. This organized arrangement makes it easier to spot any missing, extra or rearranged chromosomes. It's like putting together a jigsaw puzzle and seeing if any pieces are missing or in the wrong place. Section 9. Solving the puzzle. Karyotyping and genetic conditions. Karyotyping plays a crucial role in diagnosing and understanding genetic conditions. This process involves taking a close look at the chromosomes within our cells to identify any abnormalities or variations that might be present. By examining the chromosomes, scientists can identify abnormalities that might be causing a particular condition. These abnormalities can include extra chromosomes, missing chromosomes, or structural changes within the chromosomes themselves. For example, Down syndrome is caused by an extra copy of chromosome 21. This extra genetic material affects the development and function of the body, leading to the characteristics associated with Down syndrome. Karyotyping can detect this extra chromosome, allowing for early diagnosis and intervention. Early diagnosis is crucial as it can help in planning appropriate medical care and support for individuals with genetic conditions. Karyotyping is a powerful tool that helps us understand the intricate world of our genes. It provides a visual representation of our chromosomes, making it easier to spot any irregularities that could be linked to genetic disorders. So the next time you hear about chromosomes or genetic testing, remember the incredible journey of karyotyping. This technique not only aids in diagnosing conditions, but also enhances our overall understanding of genetics. It's a fascinating process that allows us to peek inside ourselves and unravel the mysteries of our genetic code. By studying our chromosomes, we can gain insights into our genetic makeup and how it influences our health and development. Keep those questions coming, science fans. The world of genetics is vast and full of discoveries waiting to be made, whether you're a student, a researcher,